So these are the subheadings that we are going to learn about this topic under, and I hope I'm able to give you a good idea. It's quite an interesting topic. So the sense of taste, which is also known as gustation, is a chemical sense that is stimulated by food and drink. It contributes considerably to the quality of life and is important stimulant for digestion. So taste must be distinguished from flavor, which includes olfactory, tactile, and thermal attributes of food, in addition to taste. Now, the site of taste. So the taste stimuli are detected by special chemoreceptors called as taste receptors or taste cells. The taste receptors are clustered in taste buds, which are located on the tongue, palate, pharynx, epiglottis, and upper third of esophagus. Tongue is the main site of taste detection. It contains numerous taste buds on its dorsal surface. The mucous membrane of dorsal surface of tongue exhibits numerous papillae, which increase surface area of mucosa available for taste receptors. Taste buds are located in walls of these papillae. Now the types of papillae. So there are four main types: circumvallate, fungiform, foliate, and filiform. So your circumvallate papillae are quite large relative to the others, 2 to 4 mm in diameter. They are around 10 to 12 in number, forming a single row in front of sulcus terminalis. Now, sulcus terminalis is a V-shaped groove. If you can see here on the diagram on right, it's a V-shaped groove with the apex posteriorly, which separates anterior 2/3 of dorsum of tongue from posterior 1/3. Okay, this is sulcus terminalis, and each circumvallate papilla is surrounded by a groove. Now 200 taste buds are located along sides of each circumvallate papilla. Next is a fungiform. So the fungiform papillae are bright red, flat, dot-like structures. Just have a look. These were the circumvallate in the diagram on right. And now have a look at the filiform papillae. They are bright red, flat, dot-like structures, 1 mm in diameter, located in anterior 2/3 of the tongue along edges, dorsum, and tip. This is 1 mm. I've missed 1 m here. There are 8 to 10 buds, taste buds on each papillae. Okay. Coming to the next one is foliate papillae. So the foliate papillae are transverse mucosal folds which are found on posterolateral surfaces of tongue. So posterolateral surface of the tongue, anterior to circumvallate papillae. And each foliate papilla has numerous taste buds. And the fourth type is the filiform. Sorry, I pointed to the wrong one earlier. First was the circumvallate, then was the fungiform, the second one. Then foliate is not given here, and the fourth is the filiform. So filiform are small conical projections covering entire remaining surface of dorsum of anterior 2/3 of tongue, giving it a velvety appearance. They are arranged in rows parallel to sulcus terminalis. They are not gustatory structures. That means they do not contain taste buds. However, they may play a role in breaking up food particles, and also they are called mechanical papillae, in contrast to the other three forms, which are gustatory papillae. So your filiform are slightly different than the other three. How? Because they are essentially not gustatory structures. They are in fact mechanical papillae, whereas the other three are gustatory papillae. And they don't, and what does it mean that they are not gustatory structures? It means that they don't contain taste buds. Now, what are taste buds? So first, let's learn about the structure of a taste bud. Each taste bud is a barrel-shaped structure. You can see a cluster of cells with a small opening, which is the taste pore, right here in surface, that allows substances to reach interior of the taste bud. So this taste pore allows the substances to reach the interior of the taste bud. Each taste bud measures about 50 to 70 micrometers in diameter and consists of the following cells. So which are those? Receptor cells, basal replacement cells, and supporting cells. Now let's learn a little bit about each of these kind of cells. So the receptor cells are elongate, bipolar shaped, and extend from epithelial opening of taste bud to its base. These taste cells have a short life, just of 10 days, and are continuously replaced by new taste cells differentiating from basal cells. Through the taste pore, the microvilli or the cilia of all taste cells protrude into the oral cavity and come in contact with saliva. The taste cells are innervated by sensory neurons, that means the primary gustatory afferent fibers at the basal pole. Although taste cells are non-neuronal epithelial cells, contacts between these cells and sensory cells have morphological characteristics of chemical synapses. Each taste nerve fiber innervates taste cells in several taste buds, and conversely, each taste bud is innervated by approximately 50 nerve fibers. So that was about receptor cells. Now let's talk about basal replacement cells. These are small round cells present at the bottom of the taste bud, and they are thought to be stem cells, which are continuously being differentiated into taste cells. Okay, and then we have supporting cells. So supporting cells, in addition to taste cells and basal cells, the taste buds contain supporting or sustentacular cells. Now, their innervation is through special sensory nerve fibers, innervating taste cells, which come from branches of facial, glossopharyngeal, and vagus nerves. So which is the facial nerve? Which facial nerve is the 7th cranial nerve, glossopharyngeal is the 9th cranial nerve, and vagus is the 10th cranial nerve. You need to all know that these are the three main nerves which are responsible for the sensation of taste. And we'll talk about them some more in a while. Each taste bud is, again, like I mentioned, for your receptor cells, even for supporting cells, each taste bud is innervated by approximately 50 nerve fibers, and each nerve fiber in turn receives inputs from the 5 kinds of taste buds. The tactile and temperature receptors of mouth, tongue, and pharynx are innervated by trigeminal nerve, which is your 5th cranial nerve. Now let's learn about the taste pathways, which is a very important topic, both theory and viva wise. So the taste pathway consists of 3 orders of neurons: first order, second order, and third order. So the first order neurons, the cell bodies of first order neurons innervating taste buds, innervating taste cells in taste buds are located in different ganglia of, as I mentioned to you, 7th, 9th, and 10th cranial nerves. Now from taste buds located on anterior 2/3 of tongue, the taste fibers run in the lingual nerve, which branches from cauda tympani nerve, a branch of facial nerve. So we are talking right now about facial nerve. The cell bodies are located in geniculate ganglion. If you look here, here is the geniculate ganglion which contains the cell bodies. From taste buds located, so this was about the taste buds located on anterior two-third of tongue, okay, which is the innervation from these taste buds are taken by the seventh cranial nerve. And I just mentioned the taste fibers run in lingual nerve, which branches from cauda tympani nerve, which is a branch of facial nerve. Now, what about the taste fibers from so this is the anterior two-third of the tongue. Now, what about the taste fibers from posterior one-third? They run in fibers of glossopharyngeal nerve, which is your ninth cranial nerve. Okay, and the cell bodies lie in superior and inferior ganglion. Okay, superior and inferior ganglia of this nerve here, as you can see in the diagram. Then the termination of, see here, the first order neurons, they are terminating here. Okay, and termination of first order neurons happens, and ultimately all taste fibers traveling in different cranial nerves, they join tractus solitarius, see nucleus tractus solitarius, to terminate in the nucleus of tractus solitarius. So this is where your first order neurons, their journey completes here in the nucleus of tractus solitarius. Now let's talk about the second order neurons, which will start from here. So the cell bodies of second order neurons of taste buds are located in the nucleus of tractus solitarius itself, in the medulla. And axons of the second order neurons, now this is your midline, okay? So now the axons from the second order neurons, which are also here in the nucleus tractus solitarius, they will cross the midline. See, they are all crossing the midline here to join medial lemniscus, okay, and terminate with fifth cranial nerve fibers, okay. So they will cross midline to join medial lemniscus and terminate with fifth cranial nerve fibers, which I just mentioned carries the pain, touch and temperature fibers in the ventral posterior medial nucleus of thalamus. So this is where the ventral posterior medial nucleus of thalamus is where the second order neurons are terminating after crossing the midline. Okay, and here is also the termination of fifth cranial nerve carrying the pain, touch and temperature fibers, but which we are not concerned with currently for the taste sensation, with regards to the taste sensation. So now we have completed we have learnt about the pathway until the second order neurons. Now let's go to the third order neurons. So of course now the cell bodies of third order neurons are located in the ventral posterior medial nucleus of thalamus, right? And so now the axons from here, from the ventral posterior medial nucleus of thalamus, they will proceed to terminate in the inferior part of postcentral gyrus, which is the part of sensory cortex known as the taste cortex or the gustatory cortex, as you see here. So is it clear? I hope we have got some idea. Just recapitulating, see anterior one-third, from anterior one-third of the fibers, anterior one-third of the tongue, the facial nerve is carrying the sensations from the taste fibers. So these are your first order neurons. Here are their cell bodies and they terminate here in the nucleus tractus solitarius. Then from here second order neurons are starting, which cross the midline and their axons terminate in ventral posterior medial nucleus of thalamus. Then from there itself, the third order neurons will start and their axons will project to your gustatory cortex. So this is broadly your taste pathway. Not too difficult, but very important. So let's learn some more about the physiology of taste. Now there are 10,000 taste buds which after the age of 45 years start declining in number, and that leads to blunting of taste sensation in old age. Now conventionally 4 basic types of taste sensations have been described, which you would have read about even at your school level. And we all know them to be sweet, salt, sour, and bitter. Now recently a 5th stimulus type called as umami or umami has been considered in the list of basic tastes. Now all the other taste sensations, which are nearly hundreds in humans, are assumed to result from various combinations of these 5 primary basic taste sensations. Now, in addition to the above, the associated sensation of olfaction, heat, cold, and texture contribute for different flavors. Earlier, it was believed that there are special areas on surface of tongue for each of the 4 conventional basic types of taste. So what we learned growing up was that the sweet tastes are detected at the tip of the tongue, the sour and salty tastes originate from the sides of the tongue, and the bitter tastes are sensed best at the base. However, now it is clear that all tastes are sensed from all parts of the tongue and adjacent structures containing taste buds. So this concept is no longer valid that the tongue can detect a taste sensation, a particular taste sensation only from that particular area. But yes, even now, the best sites for detection of those tastes remain the same, but they are not the only sites. The entire tongue and the adjacent structures around it which contain taste buds, they are able to detect all of these tastes. Now, let's learn about some substances which produce the primary or the basic taste sensations. So now sweet sensation is produced by a number of organic molecules, which includes sugars, glycols, alcohols, aldehydes, esters, etc. You would have heard about saccharin. It's a chemical which is 600 times as sweet as sucrose. Being non-calorigenic, it is often used as a sweetening agent for diabetic patients. Then the salty sensation is produced by anions of ionizable salts, especially sodium chloride. Sour sensation is produced by acids, and intensity of this sensation relates to some degree to pH of the stimulus solutions. The bitter sensation is produced by alkaloids such as quinine, caffeine, nicotine, and strychnine. Now, many alkaloids are actually harmful when swallowed, and so perhaps highly bitter taste has been given by nature to these substances to prevent their ingestion by humans and animals. And fifth, the latest addition to the tastes, which I just told you about, the umami. The umami sensation, which has been recently added, is produced by glutamate, particularly by monosodium glutamate, which is used extensively in Asian cooking. So this taste is pleasant and sweet, but it differs from the standard sweet taste. Now about the transduction of gustatory stimuli. So transduction of gustatory stimuli into electrical signals is initiated at the level of receptors. The taste receptors are basically chemoreceptors stimulated by substances dissolved in the mouth by saliva. the dissolved substances, they act on microvilli of taste receptors exposed in taste pore of taste buds. This interaction typically depolarizes the cell either directly or via action of secondary messengers. This causes development of receptor potential in receptor cell, which in turn generates action potential in sensory nerves. So taste receptors are chemoreceptors. Just like any chemoreceptors, the same process will be needed. The depolarization, development of receptor potential, and which in turn generates the action potential. And the mechanisms involved in transduction of 5 types of basic taste stimuli into electrical signals are different. Now, how does the transmission of information about taste happen up till the cortex? So detection of tastings into receptor potential happens that induces action potential in the taste cell and causes release of neurotransmitter at synapses formed between taste cell and sensory fibers. Now, each sensory fiber contacts a number of taste cells, and each taste cell synapses with numerous sensory fibers. Thus, the electrical activity recorded from a single sensory fiber represents input of many taste cells. So, as I mentioned, in the taste pathway, the signals carried by sensory fibers that innervate taste buds travel through several different nerves to the gustatory area of nucleus of solitary tract, which relay information to the thalamus. The thalamus then transmits taste information to the gustatory cortex. Now, how does the encoding of the taste information happen? So each sensory fiber carries information derived from a variety of taste stimuli. However, each fiber responds best to one of the 5 primary taste qualities. Thus, encoding of gustatory sensation is not simple or label-lined chemical sensory system. Instead, identity of taste stimulus appears to be encoded by a unique pattern of inputs from many separate fibers that provide components of patterns for different stimuli. Now, processing of taste information involves comparison of activity of different cells that respond preferentially, but not exclusively to certain features of sensory stimuli. So the encoding is not a simple process as one might think. It's a unique pattern of inputs from many separate fibers, and that provides the component of patterns of different stimuli. Now, taste threshold and intensity discrimination. So what is taste threshold? So to be recognized as a salty substance, its concentration needs to be only 0.01 moles. Whereas for quinine to be perceived as bitter, its concentration needs only to be 0.000008 moles. This correlates with the notion that bitter taste serves as a protective function against dangerous alkaloids, and thus its intensity is high. What is intensity discrimination? It is the ability of humans to discriminate differences in intensity of tastes, like intensity discrimination in olfaction is crude. 50% change in concentration of substances being tasted is necessary before an intensity difference can be detected. Also, women are more sensitive to sweet and salt and less sensitive to sour taste. Now, the sensation of flavors. So, what are gustatory inputs? Now, a multitude of different sensations of flavors that one experiences results from a combination of gustatory, olfactory, and somatosensory inputs. So the gustatory inputs, they consist of almost infinite varieties of tastes, which are synthesized from 5 basic taste components that we talked about earlier. Then olfactory inputs are responsible for much of what we think is flavor of foods. Volatile molecules released from foods or beverages in mouth are pumped into back of nasal cavity retronasally by the tongue, cheek, and throat movements that accompany chewing and swallowing. Although olfactory epithelium of nose clearly makes a major contribution to sensation of taste, we experience taste as being in mouth and not in the nose. Now, it is thought that the somatosensory system is involved in this localization, and that coincidence between somatosensory stimulation of tongue and retronasal passage of odorants into nose causes odorants to be perceived as flavors in the mouth. Somatosensory input frequently contributes to the sensation of flavor. This component includes texture, which is consistency, and temperature of foods, as well as pain sensations evoked by spicy and minty foods and by carbonation. Now let's learn about phenomena of variation and after effects in taste sensations. It has been reported that taste sensations exhibit after reactions and contrast phenomena. These are similar in some way to visual after images and contrasts. Some of these occur due to chemical tricks, while others are considered to be result of a true central phenomena. Now, what are the factors which influence taste sensation? So, first of all, the area of stimulation. So, perception of sense of taste is directly proportional to area of taste buds which are stimulated. Therefore, stimulation of a small area of tongue by one drop of solution produces weaker sensation than same solution by the whole mouth. Then, the temperature of tastant. An optimal response to taste-producing substances is obtained when their temperature is between 30 to 40 degrees Celsius. Then we already talked about age of a person, how that affects number of taste buds, which start decreasing after the age of 45 and result in blunting of sensation of taste. Then I already told you that there is gender disparity in how we experience taste. For example, women are more sensitive to sweet and salt taste and less sensitive to sour taste. Then adaptation. So taste sensations adapt rapidly when the taste producing substance is kept for a long time in one place in the mouth. Adaptation is peripheral, and further adaptation to one acid produces adaptation to other acids because your H ion is stimulus in all the cases. Now, interaction between taste producing substances also affects the taste sensation. For example, reduction of sour taste of fruits by sucrose is a well-known phenomenon. Then effect of taste modifying proteins. Now there is a taste modifier protein known as miraculin, which has been discovered in an West African plant. So when this is applied to tongue, this protein makes acids taste sweet. Then abnormalities of taste sensations obviously will affect various taste sensations. So what are these abnormalities that I just mentioned? So first is ageusia, which is absence of taste sensation. So the causes are lesions of mandibular division of trigeminal nerve through the lingual branch of which cauda tympani nerve reaches the tongue. So lesions of this mandibular division of trigeminal nerve causes loss of taste sensations in anterior 2/3 of tongue. So your trigeminal nerve is your 5th cranial nerve. Then lesions of facial nerve, which is your 7th cranial nerve, also leads to loss of taste sensations in anterior 2/3 of tongue. Lesions of glossopharyngeal nerve, which is your ninth cranial nerve, are associated with absence of taste sensations from posterior one-third of tongue. Then drugs like captopril and penicillamine, which contain sulfhydryl groups, cause temporary loss of taste sensation. The reason for this effect of sulfhydryl compounds is not known. Then there's also a condition known as familial dysautonomia. It is a congenital, widespread sensory disorder characterized by absence of taste sensations associated with other abnormalities, such as postural hypotension, lacrimation, hyporeflexia, and insensitivity to temperature and noxious stimuli. Then the second abnormality that we are talking about is hypogeusia. Hypogeusia refers to a diminished taste sensitivity. In it, taste sensations are not completely lost, but there occurs an increase in threshold for different taste sensations. Many different diseases can produce hypogeusia. Then, dysgeusia. It refers to a disturbed sense of taste. It is a feature of temporal lobe syndrome. Then your fourth abnormality of taste sensation is selective taste blindness. It is an inherited autosomal recessive trait characterized by markedly elevated threshold for phenylthiocarbamide, PTC, also known as PTC, a chemical substance with a very bitter taste. So such individuals are called non-tasters for PTC. This defect is highly selective. Probably there is a particular receptor protein which is not synthesized in these individuals. So this was about the theoretical aspect of taste sensation. Now I'm going to touch upon the practical aspect, because it is something that will be good that we cover along with the same topic. So in your practicals, when you are testing for cranial nerves, especially your 7th and 9th cranial nerves, though they have mixed or motor functions also, but an important function that they have is that of sensation of taste detection. And so for whenever you test those two cranial nerves, you will test for the sensation of taste. So let me take you a little bit back into the theory. There are going to be some repetitions, but we can just reinforce the concepts. So though the sensations of taste, gust means taste. That's why it is also known as the sensation of gustation, and smell are closely related. They are anatomically distinct. They are commonly called chemical sensations because they are stimulated by chemicals. And these substances are called tastants for taste and odorants for smell. They are dissolved in oral and nasal mucus. So these sensations help in avoiding ingestion of harmful foods and chemicals. That's the importance of a sensation of taste, as we learned about how bitter alkaloids are perhaps best avoided in humans and animals. Then we talked about different types of cells which are present in 40 to 80 micrometer ovoid structures called as taste buds. I showed you the diagram of that, which are around 10,000 in humans. The sensory receptor cells have vesicles and microvilli that project into the taste pore. Their bases have synaptic connections with afferent nerve fibers of 7th, 9th, and 10th cranial nerve. Vagus is your 10th cranial nerve. Then taste buds, I told you about the different papillae. They are present in fungiform, valate papillae, but absent in filiform papillae. And sensory cells are replenished from supporting cells. I had also told you that filiform papillae are mechanical papillae, whereas the others are gustatory papillae. Then we just talked about the 4 basic taste modalities which were known earlier, but now the 5th one, the umami or umami, has been added by the Japanese to the list, and it is excited by glutamate. especially your monosodium glutamate, the MSG that we have heard a lot about in the last few years, sometimes considered harmful, and the other times it has been considered to be all right for the human body. Still remains controversial though. So its taste is described as meaty or savory, and it's often used a lot in Asian style of cooking. It's naturally present also in many foods, and as I said, added to Asian foods to enhance taste. So traditionally, these are the areas which you see on the right side, the top right diagram, where each of the taste sensations were known to be felt only at these areas earlier, but now, as I've told you, the tongue can experience them throughout its structure, not just at these particular locations, but still these locations are best at identifying these particular tastes. So we knew that sweet is felt at tip, salty taste on dorsum, sour along edges, and bitter at the back. And now we know that they are experienced from all areas of oral cavity, and the pattern of stimulation of taste receptors produces a particular taste. With some foods, like for example hot sauces, there is also an element of pain, and touch, temperature, and texture also contribute to taste. Flavor is a combination of different tastes and smells. So you can see here that some foods naturally contain these tastes. Now, coming to the practical aspect, how do we test for the taste sensation? So the materials we need for that are strong solutions of sucrose, around 10%, sodium chloride, 15%, and weak solutions of acetic acid. Also quinine sulfate, 0.1%, all to be kept in drop bottles. Then you need small cotton swabs or toothpicks or gauze, or even a glass rod can be used for the same, and 4 cards with sweet, salt, sour, and bitter printed on them. So what is the procedure to test for the taste sensation? So first, I'll tell you about how we test for taste sensation of the facial nerve, the 7th cranial nerve, and then the similar process is repeated for testing taste sensation with regards to the glossopharyngeal nerve, which is your 9th nerve. The only difference between testing of these two nerves is the location, right? So I told you the facial nerve is responsible for taste sensation in anterior 2/3 of the tongue, and glossopharyngeal is responsible for taste sensation in posterior 1/3. So first of all, please take the history from the patient if there are any abnormal taste sensations, okay? Then instruct the subject not to speak during the test. This is an important precaution. He or she is asked to indicate the taste felt by pointing to an appropriate card to indicate the particular taste sensation. So we just talked about some indicator cards with different sensations written on them, printed on them. So the subject is not to speak, is not to withdraw his or her tongue back at any point, is simply to point or pick those pieces of paper to tell us what taste sensation is being experienced. Ask the subject to rinse the mouth and then dry it with gauze. Ask the subject to protrude the tongue and hold it with a swab. Moisten the swab with few drops of sugar solution, apply it to the tip of the tongue, and ask the subject to indicate, as I said, without withdrawing tongue, without speaking, the taste which is experienced. And you repeat the same procedure with all 4 substances on the anterior 2/3 of tongue. So this will complete your sensation of the facial nerve. Now for testing the sensory function of 9th cranial nerve or the glossopharyngeal nerve, sensation of taste will be checked over the posterior 1/3 of tongue, and exactly same format as what I just told you. So you can have a look here. Let's see here, they are assessing the taste over anterior 2/3 of tongue with a swab and touching the solution. All 4 have to be done one by one. We usually do the quinine solution in the end because that's a more strong effect, more long lasting. So one by one we test in the anterior 2/3 of tongue. Here you can see, here we are testing for taste sensation in the posterior 1/3 for the glossopharyngeal nerve. And here anterior 2/3 for the facial nerve. So this see this is how you note down the your findings or your inference. So as you know, both these nerves, they have other functions also, like secretomotor function. Facial nerve has also motor function. We are not concerned with that at the moment. We are only, for today, we are only talking about the test for taste sensation. But I'm just showing you how the inference is to be recorded. So here, for each taste, you will say present or absent based on what the subject shows you, which card or which taste the subject indicates to you. And similarly, for glossopharyngeal nerve, again, there are other things to check for when we do the cranial nerve testing, like a palatal reflex. Again, something we're not concerned with ourselves with today. We are only going to mention about the taste sensations in posterior 1/3 of the tongue. Again, you can mention all 4 there and mention if they are present or absent. So, important precaution, remember, as I mentioned, but is often asked and checked during when you perform the practical. The subject is not supposed to speak, not supposed to withdraw his or her tongue, and is supposed to indicate using those printed cards. Now, finally, I'm just going to one last time recapitulate the taste pathway, which I hope by now is quite familiar to you all. So, 3 cranial nerves contain axons of first order gustatory neurons that innervate sensory cells in the taste buds. The 7th cranial nerve, facial nerve, from anterior 2/3 of tongue. 9th cranial nerve from posterior 1/3 of tongue, which is the glossopharyngeal, and 10th cranial nerve, vagus, from part of pharynx and epiglottis. We don't usually test the 10th cranial nerve, the vagus nerve, for sensation of taste. You will test for it, and you will do different tests for testing of the vagus nerve. We are not concerned with that as of today. Then second order neurons. So first order neurons are here, see, from the taste bud, and then the taste fibers from 7th, 9th and 10th nerves. Then the second order of neurons, they form nucleus of tractus solitarius. See, this is the nucleus of tractus solitarius, which travels up in ipsilateral midline, medial, and travel up in medial lemniscus to end in nucleus lateralis, posterolaterales of thalamus. Here. Then from thalamus, they are relayed to the taste area 1 in post-central gyrus, and other fibers from the thalamus end in taste area 2. So in this diagram, you also learn of 2 taste areas. Taste area 1 here in the foot of post-central gyrus, and 2 here in the insula. And then further on to the limbic system and hypothalamus. And I told you earlier also, the general sensations of touch, pain and temperature are carried from oral cavity by 5th cranial nerve. So let's recall again the abnormalities of taste. Ageusia, absence of taste. Hypogeusia is decreased sensitivity. Dysgeusia is disturbed sense of taste, and with different diseases and drugs can cause these conditions. Now, what is the physioclinical significance? So, This is again something you will learn in detail later on when you especially study about facial testing of the facial nerve. You will learn what is an upper motor neuron paralysis and a lower motor neuron paralysis. But just for now, in an infranuclear lesion, that is a lower motor neuron paralysis, if involvement of fibers of cauda tympani is there, then taste sensation is lost from anterior two-third. But in case of supranuclear lesions, taste sensation is not affected. So it's only affected in case of infranuclear lesions, and that also if there is involvement of cauda tympani, which is your fibers of the seventh nerve. Then just to highlight some important questions which may be asked both with regards to your theory and viva. The pathway for taste sensation, then abnormalities in relation to taste sensation. Okay, this is pathway for sensation of smell. So no, this will come in your when you study sensation of smell. Then what is the role of facial nerve in taste? Test the taste functions of the facial nerve. You may be asked also about the glossopharyngeal nerve. And I just mentioned what precautions to be observed, which I told you not to speak. The subject is not to speak, not to withdraw the tongue, and indicate only using the indicator cards. So that's all. Any doubts, any queries? Please feel free to ask them. Yes? Do you all have any queries that you want to ask me? Otherwise, feel free to put them up on the group and I can, I'll be happy to answer them. Are there no doubts? Okay, I have a few questions here. So somebody, one of you has asked about miraculin. Okay, so miraculin, I told you, is has been discovered from a West African plant. It's a kind of taste modifying protein. And the interesting thing about it is that when it's applied to tongue, this protein can make the acids taste sweet. Okay, then see, I think the fibers from the NTS, which I told you earlier, they showed that they cross the midline, the second order exons. But I just, when I felt that the recent diagram which I showed you showed them to be ipsilateral, so I will just check this one more time. And I'll let you know in the group. I think they do cross the midline, but I'll get back to you on this. Then somebody has asked about adaptation. So adaptation is basically, see, you would have experienced this practically also when you keep a substance in your mouth in one place for a long time, then your, you don't feel that particular taste sensation so strongly, right? Because your taste buds have gotten adapted. Okay, and it's a peripheral concept, and sometimes adaptation to one acid produces adaptation to other acids also, because in case of acids, the main stimulus is the H ion. Yes, so you're right. I am finding some discrepancies in both the diagrams. So the first one I took from theory, a theory book, and the second one is from a practical book. I will just check on this and let you know which is the correct one for your reference. Okay, don't worry, I'll share it in the group. So I think I have answered most of the questions which have been put up by you all. If there are any more, uh, you please feel free to put them up in the group. Okay. Which book to be referred from practical aspect? Okay, so I referred to multiple books. One book is which is by written by Dr. C.L. Ghai, which is a good book for your practicals. Another book is which has been written by our HOD sir, Dr. Varshne, and director Professor Dr. Mona. So that's another one. And I also referred to the practical book written by Dr. A.K. Jain and Dr. Chatterjee. So these are the books I referred to, but I think if you all just pick any one of these, like maybe Dr. Varshne's book or one by Dr. C.L. Ghai or Dr. A.K. Jain, any one book is good enough to give you most of the information that you need. So, are there any other doubts? Yes, shall we end the session for today? Okay, so I'm not hearing anything back. And which book should we read for CNS? See, the best books for you all are, I would say Guyton is the best book. And also you can read a bit of, add some extra information from Ganong if you want to really enhance your knowledge and your answers. Any other query? See, about one of you has asked about the loss of taste sensation in COVID. I'm not too sure about the entire physiological process at the moment, but I think it may be because also that your sense of smell is also affected, and you know that we mentioned that the sense of taste and sense of smell are quite intricately connected. In fact, I think I'll just show you in the first few slides itself, I mentioned that the sense of taste, see here in this slide, so the site of taste is definitely in the tongue, but the olfactory, see for any flavors, your olfactory, tactile, the thermal aspects also attribute to sense of taste, right? And even retronasally, the odors, they contribute to your sense of taste. See, the associated sensation of olfaction, heat, cold, and texture contribute to different flavors. So I think because, and see, we also talked about the olfactory inputs, didn't we? Which are very important for flavors of foods. When we said that the volatile molecules are released from foods or beverages in mouth, and they are pumped back into the nasal cavity retronasally by the tongue, cheek, and throat movements which accompany chewing and swallowing. Right? So basically, there is a huge role of olfactory epithelium of nose. It makes a major contribution to sensation of taste. Still, I mean, we experience taste as being in mouth, not in nose. So if your COVID is impacting a sense of smell, which is nearly 95% of the time, there is a sense of smell. So then it is jointly associated with the loss of taste. And you can think of it as even when you have a common cold, right? So your sense of smell is affected and you can barely taste the food that you're eating. So I think it would be a similar phenomena that would contribute to loss of taste in COVID. So that answers, I think, all the queries that I have received from you all. And as I said, if there are any more, please feel free to share them in the group and I'll be happy to answer them. Okay, what is this about? Another one has come up. Vagus nerve does what? So vagus is your 10th cranial nerve, okay, and it has various functions, but they are not. I mean, it is a complex nerve. It's a mixed nerve. It also contains parasympathetic fibers. In fact, it is one of the longest and most complex nerves, which runs from the brain through the face and thorax to the abdomen. So, I mean, it has a lot of functions. It's practically one of the most important nerves. It has cardiac, esophageal, pulmonary branches. So, vagus nerve testing is in fact one of the extensive cranial nerve testings that you will do, and you learn about it when you do the cranial nerve testing. But here for the taste sensation, vagus nerve is not generally tested for taste, although it is responsible for contributing to the taste sensation, but we don't test it at our level. We don't test it during testing for taste sensation. Okay, so with that, I think we can end this session. Thank you.